Today we are going to study TS of Anther. Before going to start this concept, we have to know what is a stamen. The stamen is the male reproductive organ of the flower. Typically, it consists of two distinct parts, a lower sterile part, the filament, and the upper fertile part, the anther. The sterile median part of anther is known as connective. The anther shows great variety in form, size, and color. Typically, it is two-lobed and the lobes are called anther lobes. Such anthers are described as dithecous. Each anther lobe consists a pair of pollen sacs or microsporangia. Thus, characteristically, each anther has 4 microsporangia. The anther wall. The mature anther wall is made up of 4 layers. Epidermis. This is the outermost layer of the anther wall. The cells of epidermis is made up of tangentially elongated and flattened cells. The epidermis performs its usual protective function. Endothecium. This is the subepidermal layer of the anther wall. Mostly it is single layered, but sometimes it may be several layered. The cells of endothecium are radially elongated and attain their maximum development at the time of anther dehiscence. The radial and inner tangential walls of these cells have characteristic fibrous thickenings. These fibrous thickenings do not develop in the region of stomium. The presence of fibrous bands, differential expansion of outer and inner tangential walls, and hygroscopic nature of endothelial cells all combine to bring about the dehiscence of anther at maturity. In some Clistogamous species where flowers do not open and in the members of Hydrocaritaceae, the fibrous bands are altogether absent in endothecium. The endothecium of those anthers which dehisce by apical pores also lack thickenings. Next, middle layers. Inner to endothecium, 2 to 3 layers of parenchymal cells are present. These are known as middle layers, as they lie between endothecium and tapetum. The cells of these layers are ephemeral and degenerate almost completely before the pollen mother cells undergo meiosis. The middle layers may store food material in some taxa, which is utilized by pollen mother cells. Next. Tapetum, the innermost layer of the anther wall. It surrounds the sporogenous tissue and attains maximum development when the microspores are in tetrad stage, after which they collapse. Generally, tapetum is single layered, but it may divide periclinary and become biseriate and multiseriate. The cells of the tapetum are relatively large and centripetally extended. They have dense cytoplasm and prominent nucleus. Cells may be multinucleate or polyploid and are comparatively rich in DNA. Tapetum usually is homogeneous layer surrounding the sporogenous tissue. In some taxa, however, dimorphic tapetum is present. Example, Nigella damascena. In these plants, the tapetum cells present towards the protuberant side of the anther are relatively small and it's called P tapetum, and the tapetum cells towards the connective are large and are called C tapetum. And there are 2 types of tapetum. They are amoeboid tapetum and secretory tapetum. And these 2 types of tapetum are seen in the angiosperms. So, in detail about the tapetum and types of tapetum and their functions we'll see in the next video class. Thank you.